Hello, my name is Yuta Takagi, and this presentation will be on using digital life simulations to examine the evolutionary origin of cellularity and metabolism. In this research, we designed and coded a digital life system to study evolution and used this simulation to study the origin of cellularity. First, a bit of background. We know that the last universal common ancestor was already cellular because cell membrane proteins such as signal receptors and the ATP synthase motor are universally present across the known tree of life. However, almost all origin of life hypotheses suggest a non-cellular beginning, such as the RNA world hypothesis, where the first entities were self-replicating RNAs, or in the autocatalytic network hypothesis, which more closely resembles complex chemistry. This necessarily means that at some point in early life, there was a transition from non-cellular to cellular entities. Our work was motivated by the fact that, even though there has been a lot of previous research on the biochemistry of early cellularity, there has been very little research examining the evolutionary causes for such a transition. I will now give a simplified explanation of the simulation. The simulation consists of a population of replicators referred to as organisms in a shared environment containing food. The environment is capped at a certain population size. Each organism has a genome composed of metabolic and cellularity genes. Metabolic genes represent logical not-and gates that denote a Boolean logic gate network. The food in the simulations are Boolean logic puzzles that give energy as a reward depending on how well they are solved by the genome's logic gate network, a property we have termed metabolic proficiency. Energy is stored in the organism and is used to read through the genome, where each step corresponds to one gene denoting a calculation in the Boolean logic gate network. The genome can read values from the puzzle's input register, process the values through its logic gate network, and rate the solutions to an output register, which is then compared to the puzzle's solution register to determine the energy reward based on the number of correctly solved digits. If stored energy runs out, the organism is dead. Cellularity genes are logically neutral in the Boolean gate network, but instead determine an organism's level of cellularity. Cellularity is defined as a value between 0 and 1, with 0 representing non-cellular and 1 representing completely cellular, in other words, entirely closed off. Cellularity genes confer cellularity according to the equation 1 minus 1/2 to the x, where x is the number of cellularity genes in the genome, asymptotically approaching 1 with higher numbers of cellularity genes. Organisms with low levels of cellularity are more open to the environment and are subject to losing or gaining genetic material or free energy from the environment, while organisms with high cellularity are more closed off. The organisms replicate their genomes and split into two new identical entities when a certain level of excess energy is reached, with each new daughter organism receiving half of their parents' energy reserve. Organisms are constantly subject to low levels of mutation in the form of small alterations to the genome, through insertion or deletion of metabolic or cellularity genes, or through small reconfigurations of the metabolic logic gate network. Organisms are also subject to loss or gain of potentially large sections of genetic material through horizontal gene transfer, depending on their level of cellularity. This form of mutation can cause drastic changes to the metabolic network, and we have found that this is almost always deleterious to the metabolic proficiency of an organism. The simulation is quite far abstracted from biological life, but it does represent a system capable of real evolution, where cellularity and metabolic proficiency are dependent variables, allowing us to empirically address broad evolutionary questions. After development of the software was completed, we needed to narrow down the parameter space to conditions that allowed for organisms to survive and evolve. During this stage, we noticed that the amount of free energy in the environment had an effect on cellularity. This observation led to our primary hypothesis. that non-cellularity will be selectively advantageous when simple sources of energy are freely available in the environment, and conversely, that high cellularity will be selectively advantageous when only complex food sources are available in the environment. In order to test these hypotheses, we ran a series of selection experiments altering the organism's starting cellularity and the amount of free energy in the environment. Simulation sets A and B began with 0 cellularity genes, giving initial cellularity values of 0%, while sets C and D began with 3 cellularity genes, giving 87.5% cellularity. Simulation sets A and C were provided environments with unlimited food and unlimited energy, while sets B and D had unlimited food but limited energy. Each condition was run with 3 replicates each, under 3 maximum population settings of 500, 1,000, and 10,000. We found that regardless of the starting cellularity, organisms consistently evolved towards low cellularity under high environmental energy conditions, and high cellularity under low environmental energy conditions, confirming our hypothesis. We further saw a corresponding trend towards low and high metabolic proficiency, with poor solutions evolving under high environmental energy conditions and good solutions evolving under low environmental energy conditions. We statistically confirmed this correlation between cellularity and metabolism, showing that they are indeed co-evolving, with the population averaged values for simulation set B taken at 250 step intervals showing a linear regression with a p-value under 10 to the minus 24. This seems to make sense mechanistically if you think about it. When easy-to-use resources are plentiful in the environment, it makes sense to be more open to that environment, and there is little selective advantage for complex metabolism. Moreover, large and consistent genomic alterations prevent the Darwinian evolution of complex metabolism. On the other hand, in environments lacking such resources, there is a high selective advantage for complex metabolisms capable of utilizing difficult-to-use resources. This provides selective pressure for a more closed-off entity, both in order to retain the metabolic products of that complex metabolism and to maintain genetic fidelity by restricting horizontal gene flow. I believe this also presents a mechanism for a Darwinian transition, where the availability of free energy drives a change in cellularity that is coupled with replication fidelity, which is a necessary condition for the evolution of metabolism. Although, full disclosure, this interpretation is still a point of debate between my research collaborators and myself. So to summarize, most scholars agree that life began in a geochemically rich environment, whether that be hydrothermal vents, tidal pools, or any manner of prebiotic soup scenario. Our research adds to the understanding of life's origins by showing that cellularity likely arose in response to a change towards a less rich environment, perhaps through some geological event, or perhaps simply through the depletion of initial reserves as life expanded. Our simulation research suggests that any geochemically rich origin of life setting would have prevented the evolution of cellular life. Replicators with access to plentiful prebiotic precursor molecules have an evolutionary incentive not to partition themselves from these resources. We also found that cellularity and metabolism would have co-evolved in response to resource limitation. Cellularity provides a replicator greater protection from mutation, allowing for a complex metabolism to evolve, giving it access to previously non-useful resources. Cellularity also prevents the products of metabolic processing to be lost to the environment. This gives a strong evolutionary incentive for complex metabolism to evolve when easy-to-utilize resources are exhausted, which in turn applies an evolutionary pressure towards concurrently evolving cellularity. These mechanisms suggest that the evolution of both metabolism and cellularity in early terrestrial life would have occurred in response to a change in the geochemical setting of life's origin, from an environment rich in prebiotic precursor molecules to a poorer environment. While any number of mechanisms could explain a change in the geochemical setting of early life. A simple scenario suggests itself wherein an initially rich environment becomes depleted as replicators grow in population, driving the shift towards more complex metabolic processes and cellularity. Lastly, I propose a mechanism for a shift from a non-Darwinian to Darwinian system, a so-called Darwinian transition. Cellularity provides a replicator with greater protection from mutation, with low mutation rates being a necessary condition for Darwinian evolution of the genome to occur. As such, the very capacity for the genome to evolve in a Darwinian manner may be subject to selection through cellularity, and by extension the plenitude or scarcity of resources in the environment may present a driving mechanism for a Darwinian transition. In conclusion, we found that cellularity and complex metabolism are not an inevitable outcome of evolution, but rather they are linked traits that are reversibly selected for by the environmental availability of resources. Cellularity and complex metabolism in early Earth-based life likely evolved in response to a changing geochemical environment and facilitated life's capacity for Darwinian evolution. Thank you for your attention and interest in this work. If you are further interested, this research was published in greater detail in the Journal of Molecular Evolution. If you're interested in using the simulation or contributing to the software, it is publicly available on GitHub. If you have any further questions or comments, please feel free to contact me at yutaatakagi@gmail.com. I would like to acknowledge my collaborators in this research, Diep Nguyen, Dr. Tom Wexler, and Dr. Aaron Goldman. This research was enabled by the Oberlin College Department of Biology, and funding was provided by the National Aeronautics and Space Administration, as well as the National Science Foundation. Thank you.